My name is Dr. Lorrie Kirshenbaum. I'm from the Institute of Cardiovascular Sciences in Winnipeg, Manitoba, and I'm delighted to be part of the faculty for the International Academy of Cardiology. The topic of my research and presentation today is going to be about the crossroads of how the endoplasmic reticulum and the mitochondria interact to regulate cell death in cardiac myocytes. So the premise of my research has largely been focused on the inability of cardiac muscle cells to replicate to any great extent after birth. And despite the prevalence of cardiac progenitor cells that have really been shown over the past 10 years to generate new cardiac muscle, the limited ability of these cardiac progenitors has really resulted in the fact that cardiac cells die after ischemic or hypoxic injury. And so therefore the focus of my research has really been trying to understand the signaling pathways that underlie the cell death pathways in the heart, with an eye toward developing therapeutic interventions that could mitigate or prevent excessive cardiac myocyte loss after ischemic or hypoxic injury. So several years ago, my lab was instrumental in cloning a protein called BNIP3 as a BCL2 interacting factor. And BCL2 is a family of pro and anti-apoptotic genes that were initially identified in cancer cells that either confer cell death or survival. And they basically program a given cell and basically tell the cell when to live and when to die. And the gene that we cloned or identified basically is activated selectively in cardiac myocytes under hypoxic or ischemic conditions. So in relation to the other family members that would otherwise promote either survival or cell death, this particular gene is switched on in a very predictable and regulated manner. And what we found, though, is that while this gene is switched on, its protein predominantly localizes to the mitochondria of cells, where it disrupts mitochondrial function. And one of the key elements with respect to understanding the cell death mechanisms that's really been elucidated over the past 10 to 15 years is a mitochondrial regulated form of cell death. So the mitochondria that we generally think about as generating ATP, the mitochondria also regulate cell death. And this particular gene that we identified, once activated, actually targets the mitochondria and disrupts mitochondrial function, activating this mitochondrial death pathway. We've developed interventions that prevent the activation of this gene or preventing it from targeting to the mitochondria and actually preventing heart cell death. So the proof of concept really is that this gene is really an interesting target potentially because it identifies a potential molecule discovery-based approach that we could use to prevent heart cells from dying. In our quest to trying to understand how this gene works, we identified a naturally occurring splice variant of this particular gene that's activated only under hypoxic conditions. And in response to stress, not only is the parent gene turned on that targets the mitochondria, we discovered that the small or the splice variant of this gene is actually targeted to the endoplasmic reticulum, where it converts cell death into survival signals. So hence we have one form of the protein targeting the mitochondria and the splice variant of the gene actually targeting the endoplasmic reticulum. And by disrupting the small gene or this new protein that we identified from targeting to the mitochondria, you actually promote more cell death. So clearly, the ability to regulate cell survival and cell death is very complex, and there's crosstalk between the endoplasmic reticulum and the mitochondria. And the discovery of the small splice variant of this protein called BNIP3 abrogates or prevents cell death from occurring under normal conditions. And our goal now is to try to develop therapies that enhance the level of this splice variant's expression as a way to abrogate or prevent cell death from occurring under ischemic or hypoxic conditions. We think the work is incredibly exciting and very novel, and I would hope will help in the next several years in helping folks or patients with ischemic or heart disease.